All right. Happy COVID-19, people. This is going to wrap up the lecture on the eukaryotic genomic control and then some of the funky genetics that applies to just some kind of random things that can happen with genetics. All right, so we left off last time. We were talking about epigenetics. We talked about methylation and acetylation, how methylation will cause the genes to condense so they can't be transcribed, and then acetylation is where the acetyl groups bind to the histone tails, causing the DNA to unwind, allowing for transcription to occur and for gene expression. Another thing that can affect the transcription of a gene is the availability of the transcription factors. And so we know that transcription factors first have to bind to the Tata box before the RNA polymerase can join and do the transcription. However, transcription factors, being proteins themselves, have to be made, and some of them actually have to be guided in to the Tata box for binding. So there are some control elements that are upstream from the transcription unit, and so you have enhancers that will bind and cause the DNA to bend, and that will guide in different transcription factors. We see in this slide that you have the enhancer region here, and when the activators bind, you have this DNA bending protein guiding our transcription factors in. They bind to the Tata box, then the RNA polymerase can bind, and then you can have transcription. Another thing that affects this is whether or not you have had the proper transcription and translation of your transcription factors themselves. So this can go back to the signal transduction pathway. So you get your signal that causes your transduction, and then the response to that signal could be the production of transcription factors, or the transduction pathway itself could activate transcription factors, and then you could have gene expression. Other things that can affect gene expression are going to be some post-transcriptional controls. So this doesn't affect how the DNA is transcribed, but it determines whether or not we can go from mRNA to the proteins. So one of the main mechanisms for this is going to be alternative RNA splicing. And so you've got your introns and your exons, and what can happen, as we see in the next picture, is that you've got all of your DNA right here that is transcribed into your mRNA. But then based on what exons are removed with the introns, you can have different mRNA sequences. And so from the same gene, you can actually have multiple polypeptides being transcribed. So this goes against the one gene, one enzyme idea, the one gene, one protein idea, and even the one gene, one polypeptide idea. So now we can begin to see how the relatively few genes that we have, 20 to 30,000, can actually possibly express for a far greater variety of proteins than we thought. Other things that affect gene expression will be how long the mRNA lasts. So the prokaryotic RNA is only going to last a few minutes, and that is because it doesn't have that 5' cap or that poly A tail. So you've got enzymes that rapidly break down the mRNA, so it's no longer functional. So this is one of the reasons that prokaryotes will, as soon as transcription starts, translation will start, and you will have multiple ribosomes binding to the mRNA even before it's done being transcribed. But they will have to do a constant generation of mRNAs because those mRNAs are not conserved. Eukaryotes, because of that 5' cap and that poly A tail, they can last for a much longer period of time, which allows the eukaryotic cell to spend more energy on building proteins than it does on building mRNA. We also, going back to our microRNAs and our small interfering RNAs, these are tiny pieces of RNA that will bind to a regular mRNA and basically create a roadblock for the ribosome. So we see in this picture, we've got our microRNA right here. So you had a double-stranded RNA that gets broken down into your microRNA. This binds to some segment of your regular mRNA. And now, because you have this double-stranded piece of mRNA, your ribosome comes and it runs into that, and it cannot finish processing and making the protein. One way that this has been applied, even though it's rather impractical, is they have been able to genetically engineer blue roses. So normally, the central vacuole of a rose flower cell is very acidic. So the pigments that are generated by the cell, when they enter into the central vacuole, they are pH sensitive, so the acid causes those pigments to be pink. So scientists thought, well, let's try to make the roses blue. So they took the gene from another flower, which is called the delphinium, which is a very, very true blue flower, not like a purpley blue, but like actual blue. And they took that gene and they inserted it into the rose plant. And they thought, well, now the rose will be blue. But the delphinium pigment is also pH sensitive. So even though that protein was being expressed and that pigment was being made, the flower was still pink. So what they did is they used some microRNAs to silence the transcription of the proteins that made the central vacuole acidic, probably some kind of proton pump. I don't know about that for sure. So that the central vacuole was no longer acidic, and then they were able to make blue roses. Yay, microRNAs. Okay, so some weird things that happen in genetics. So what we're going to talk about here very briefly are what are known as transposable elements or transposomes. These are jumping genes. And so as we see in this picture, you've got this gene that makes a protein called the transposon enzyme or transposonase, and that causes the gene to get cut out and then inserted somewhere else on some other chromosome. And what that will do, that will disrupt whatever gene it was inserted into, and then that gene is non-functional. So they're called jumping genes. If you've ever seen the corn that people use in the fall to decorate that has all the multicolors, all those multicolors result from different areas where these jumping genes enter, and they disrupt the way that the colors are expressed, giving you multicolored corn kernels. These are different than plasmids. Plasmids exist independently as their own separate little circles, but these are always inside of a chromosome. These exist in prokaryotes and in eukaryotes. Typically, this is just explaining how it works, which I already did. Typically, in a prokaryote, you have what's known as an insertion sequence that simply either makes a copy of itself and inserts it somewhere else, or cuts itself out and inserts it. You can also have what are known as true transposons, which are some gene in between two insertion sequences, and so that will cause it to jump and move around. The actual mechanism of how that works isn't super important. Just know that they can cut themselves out and move them, copy themselves, and the copy insert somewhere else, or you can have something that's known as retrotransposition using an enzyme called reverse transcriptase. So transcription is DNA to RNA. And so what reverse transcriptase does is it reverses. So it takes RNA that has been transcribed and then it copies it backwards into DNA, reverse transcriptase. And then this DNA becomes the thing that gets inserted. So we see that here we have the DNA that gets transcribed, then that gets reverse transcribed into DNA, and then this gets inserted into some other chromosome somewhere. So the problem with transposons is they're generally pretty random of when they happen. So that will disrupt a gene. If it's a prokaryote, it can transfer genes around like antibiotic resistance. If that gene in between the two insertion sequences is copied multiple times. What's up, buddy? You want to come say hi? Here we go. Everybody say hi to Milo. This is the guy that likes to wake me up in the middle of the night for no reason. All right, so it could copy multiple times, and then you could potentially have a gene that is amplified and copying extra proteins. Maybe it's a growth factor, maybe it's a promoter, and then you could have over expression of a gene that could cause cancer or cell death or something. So cancer then, as we know, is uncontrolled cell growth. This could happen multiple ways. What are you doing? Hello. Yes, don't step on the keyboard. All right, so the cell cycle is controlled by proteins and enzymes. So the overexpression of some of these enzymes could change the way that the cell grows. And this could be because you have extra promoters being made. Maybe you have a gene that makes the promoter being made in an extra. You're being very distracting. You are not helping the cause. Don't step on the keyboard. You're being a bad kitty. You need to go. Yeah, hi. I see you. Go away. All right. So, okay. So the mechanism for cells to become cancerous, very generally, because all the mechanisms are not even known. One of the mechanisms is for a cell to start expressing what is known as an oncogene, which is a tumor-causing gene. Prior for a gene to become an oncogene, it's known as a proto-oncogene, or has the potential to become one. So an example of a proto-oncogene is going to be the BCRA gene, the BRCA1 gene, which is a tumor suppressor gene that normally blocks the production of tumors in breast tissue. So if you have the mutated form of this, you have a greater probability of developing breast cancer. If you have two versions of the mutated form, then you're even more likely to develop breast cancer. Does it mean you will? No. And even if you have two correct versions of the BRCA gene, doesn't mean you won't develop breast cancer either. So that's what this video is about. You won't be able to watch it through this, but you can look it up. The Cancer We All Have. It is a TED Ed, and it is very good. Changes to a proto-oncogene that could lead it to becoming cancerous. You get a new promoter, so then maybe a growth factor gets copied extra times because the new promoter is more active. Maybe a gene gets copied incorrectly, maybe in crossing over, and then you have 2 of a specific gene in a cell, and that causes the growth factor to be produced more. Maybe there's a point mutation, it makes the mRNA last longer, changes in the tumor suppressor, anything like that. So here we see a new promoter causing overexpression of a gene product. Same thing with gene amplification, point mutation. Maybe this makes the transcription factors bond longer. Or maybe you have that mRNA just lasting a lot longer, making some product that then promotes cell growth to be faster. Then you have less time for proofreading DNA, then you get some mutations, and then the cells become cancerous. Different causes for this. It could be chemicals such as pesticides or the hundreds to thousands of them that are in cigarette smoke. Don't smoke, it's bad for you, it's dumb. It's also super expensive, not only to you, but to the healthcare system. All right, radiation, some of that we can avoid. We put large lead vests on our chests when we're at the dentist. Other radiation you cannot avoid because it's coming from the solar system. Viruses, such as the HPV virus, that can cause cervical cancer. Other viruses, they get in there, they can disrupt genome and control systems. The random jumping of genes into other genes can trigger cells to become cancerous. Errors in mitosis and meiosis. As you get older, you get more and more mutations. Your cells lose the ability to correct those mutations, so it becomes more and more likely that you could get cancer. And then you have those epigenetic factors based on what you have been exposed to in your lifetime, what your mother was exposed to, potentially going in and modifying your genes. One type of tumor suppressor can be highly methylated, and there are some demethylators in green tea that actually can help reactivate tumor suppressor genes, and I think that's one of the reasons why green tea can help prevent cancer. All right, so that is it for this. Hopefully that was understandable. Not a ton of this will be on the test, which will be on Monday, fingers crossed. So email me if you have any questions. Thank you.